Hello, let's see how we can answer some GCSE Biology exam questions on cells and microscopy. We're going to look at the structure of some different cells, how to set up and use a microscope, and how to calculate its magnification. This question is all about cell structures. We have to draw a line from the structure or organelle on the left to the matching cell type on the right. There are some important key terms here that we need to understand before we can answer the question. Prokaryotic cells are bacterial cells, so they have no nucleus. Eukaryotic cells are the plant, animal or fungal cells, and these do have a nucleus. If we look at the diagram of these cell types, we can see which ones contain the different structures. You can see the nucleus present in the animal and plant cells. The vacuole is shown here as this large, yellow, fluid-filled sac, and the plasmid is a piece of DNA found in bacterial cells. Remember that bacterial cells don't have a nucleus. They have their DNA just loosely found in the cytoplasm as circular chromosomes, or as plasmids. I bet you probably already know how the nucleus communicates with all the other organelles, don't you? By cell phone? If we look at some of the other differences between prokaryotes and eukaryotes, we can see that prokaryotes have no mitochondria or chloroplasts. In fact, mitochondria and chloroplasts are similar in size to bacterial cells, so clearly they're not going to be present. Prokaryotes have smaller ribosomes. They do have cell walls, but they're not the same as those found in plant or fungal cells. Animal cells, of course, don't have cell walls. So going back to the question, we should now know that the nucleus is only found in eukaryotic cells. The plasmid, as we've just seen, is found in prokaryotic cells, and the permanent vacuole is only seen in plant cells. And finally, criminals are usually found in prison cells. This next question asks us to name the structures A, B and C in the plant cells shown. If we put the diagrams back up, we can see that the structure A must be the vacuole. Structure B is a bit difficult to identify because it's so small, but we can safely say that it's not a vacuole or a chloroplast, but it could be a mitochondrion or a ribosome. Structure C around the outside of the cell is clearly a cell wall, which makes structure B a ribosome, and we can tick the bottom box. The next question is about setting up a microscope to look at some onion cells. The student saw some cells on slides A and B, but they were not very clear. The first question asks how the student should adjust the microscope to see the cells on slide A more clearly. Those cells don't look to be in focus, do they? If we show a labelled diagram of the microscope, we can see that it's the fine focusing knob that needs to be adjusted. The second question asks how the student should adjust the microscope to see the cells on slide B more clearly. This time the image is too small, so we need to change the objective lens to a higher magnification. So just make sure that when you write your answers you are absolutely clear about what you're saying. Use the key terms and the correct names for the microscope parts. You'll need to learn these labels on the microscope as well as how to use them. This last question shows a drawing of one of the cells seen by the student. It gives the length of the image at 112 millimeters. It also tells us the length of the actual cell at 280 micrometers. It then asks us to calculate the magnification of the drawing. The equation we need to answer this question is magnification equals image size divided by actual size. Now personally I don't like these formula triangles, but I know that some of you do find them easier to remember the formulae. This one is particularly easy to remember because it says I am. Shush! Using this triangle then, if you want to know the magnification, just cover the M with your finger. Then you're left with the image size over the actual size as the equation states. So now we have the equation, we just plug the values into it from the question. But before we carry out the calculation, we absolutely have to make sure that the units of the measurement are the same for each dimension. We can see that the image size is in millimeters, but the actual size is in micrometers, and it's really important that we convert one of these units so that they're both the same, otherwise the equation won't work. So how do we do that? We can either convert the 112 millimeters into micrometers by multiplying by 1000, or we can do it the other way around and convert the 280 micrometers into millimeters by dividing by 1000. So let's convert the 280 micrometers into 0.28 millimeters. Now the units are the same, we can complete the calculation. We've got 112 millimeters divided by 0.28 millimeters, which gives us the magnification of 400 times. If you need to replay this last part to go over it again, please do so. I know it does get complicated. So that's it. Please make sure you stay in touch. You can get some great science content, updates or plenty of revision tips by following me on plutoniumscience.com or via my Twitter at PU94Science. If you prefer, you can email me directly. Keep up the revision and good luck with the exams. Take care.